Well, hello, all you beautiful people. I'm really excited to be here today to talk to you in one of our camp's space talks. I'm going to turn the direction around a little bit with my talk. I'm still going to be talking about space, but we as macroscopic humans tend to think about space in the macroscopic. We tend to think about our surroundings. When we are thinking about astronomy, we are looking outwards into the cosmos, and we are trying to understand that space. Well, what I do is I delve into the microscopic space, and it is through the microscope and through the field of microscopy that we can look inward or, inward towards the complexity of life and the near infinite complexity and beauty of biology and of the intersection of chemistry and biology and physics, which are really just a matter of scale. So my talk today is going to be about microscopes in the field of microscopy. My name is Dr. Nathaniel Peters. I go by Sunflower on the playa, and I work at the University of Washington training scientists how to use 3D laser scanning microscopes. So these are microscopes that, similar to how an MRI can slice through a body with magnetic fields, we are slicing through cells and tissues with light. And we are then able to reconstruct those cells and tissues and learn about the complexity within. Now, I did hear a few words talking about how we have a pretty good, maybe a pretty good understanding of what lies within. I would counter that we have an understanding of the language. Maybe we have the words, some of the paragraphs, and a few of the pages, but we are nowhere close to understanding the complexity within, and at a biological level. And furthermore, every time we think we have it figured out, we think we have an understanding of that scale, the tables turn and another level of complexity arises. And it's truly, truly beautiful. So I'm going to get started here. What I'm going to do today is take you on a journey through where microscopy started, where microscopy is today, and with that, some of the most exciting new innovations in microscopy that are allowing us to look at life in a way that we never really believed possible. One of those principles and one of those hurdles to overcome is the fact that when you are measuring things with light, light has a wavelength. And let's say we're talking about visible light in between about 400 and 700 nanometers. Well, if you are using something that is 400 to 700 nanometers in size to measure things, you really can't discern or resolve anything smaller than about half that size. So for many, many years, our limitation when we are looking inward in its cells and tissues has been about 200 nanometers, or 0.2 microns, or 2/1000ths of a millimeter. Wait, 1/5000th of a millimeter. Well, molecules are much smaller than that. Single protein subunits that make up many of the parts of our cells may get down to something like 5 and 10 nanometers in size, maybe even smaller. So there's really been a limitation at what we can do up until very recently. So let me take you on this little journey and mirroring. Yeah, great. Okay, cool. So let's get started. Where did microscopy begin? Well, Robert Hooke in the 1600s invented what we consider to be the first light microscope using standard light from around us in the world to magnify organisms and to magnify things that he was looking at. Ramon Cajal was one of the first influential scientists who injected neurons in the brain with fillings that allowed him to see the beautiful and intricate structure of all these individual neurons. Alexander Fleming, who is best known for the discovery of penicillin. He was also a microscopist, and he was one of the first people to look at cells under a microscope and to see these things which we now know are chromosomes that contain our DNA, and to watch these divide. So this was in the early 1900s that we were first able to look at cells and see these kinds of processes. However, we only could see what overall was happening. We didn't really know what we were looking at. We didn't know what all the pieces were. And that's where then, we spent many years like that. So discoveries were made. You might think about a light microscope. Okay, are we? Oh, no. Okay, that may not play. We may not think about a light microscope in terms of like pond water samples, things you may have done in elementary school or high school. There were innovations in polarized light microscopy that allowed us to see the texture of things within the cells, to allow us to see the nucleus where the DNA is kept. But still, we were very much just scratching at the surface. We were able to see some other beautiful things like this. We knew that the life had these incredible properties. This is a sea urchin embryo demonstrating a property that we call birefringence, which is basically a result of refraction of light. And as you can see, as this embryo turns, the light changes. Well, this embryo has a skeletal structure that is basically a crystal. And the only way this can happen is through the growth of a crystalline structure. So again, these were some of the things that we could see. They were beautiful, but we didn't have a lot of understanding as to what they were or what they were doing. Then came the discovery of fluorescence and fluorescent proteins. And I like to think about a quote from this. Am I going here still? Okay, can everyone hear me? All right, great. I like to think about a quote from the Lord of the Rings from Galadriel saying when she gave the light of the even star to Frodo, may this be a light for you in dark places when all other lights go out. Well, what we have here is the structure of what we call green fluorescent protein. This was isolated from jellyfish in the Puget Sound of Washington, and once its genetic code was understood, scientists began inserting this into model organisms and learning how to light up certain structures. And it was in this way, in the context of darkness, only lighting up specific things, that we were able to watch those specific things and what they were doing and start to get all these pieces which we're now trying to assemble. This is just a cute little example of green fluorescent protein in an organism. This is my GFP albino axolotl named Aria, who lives in my office. But you can see that through an integration of a GFP transgene, or basically an insertion of a GFP coding sequence into her DNA, her muscles now light up under blue light when we shine blue light on her. These tissues that have been integrated with this fluorescent construct glow green. So this transformed the field of modern microscopy. We then started to light up all these small structures. We then learned the limits of what we could do. I had talked to you about the limits of resolution, about the limits of measurement. Basically, at a certain point, if you're thinking about measuring with a meter stick, and you have, in a modern meter stick, you have centimeters and millimeters. At this point, we were really at the centimeter stage. So we were able to see things, certainly got an improvement over the meter stage, but we were still, it became very blurry and distorted when we tried to look really, really small. So scientists first attempted through computation, through algorithms, to learn about the properties of points of light, how they behave when they transform going through the lenses of a microscope, and be able to teach a computer program how to deconvolve or basically de-blur these light structures so that we could get a bit of an improvement. but it still wasn't close to our ultimate goal of getting down to single proteins and molecules within these cells. Now this leads to what I do, which is called confocal microscopy. I train scientists how to use these laser scanning instruments that can perform three-dimensional imaging of cells and tissues. In short, we use a very focused point of laser light to scan a specimen, like a laser printer prints a page or a laser scanner scans a page. We scan our tissues and cells in prepared samples. We use a little optical trick. Really, it's just a tiny little pinhole in a piece of metal that's adjustable. But when we align it at the right place in the light path of this microscope, what we can effectively do is block all the out-of-focus light. So let's say you're focused on me at the moment, or I hope you are. You see me, but you also are taking information in as to what is in front of you and what is in front of me and what is behind me. That information is out of focus, but if you have things in the way, they're going to obstruct your vision of me. What we do with a confocal microscope is block everything that is in front and behind so that you would only see me. In fact, maybe you would only see the surface of my eyes. That's the kind of thing that I do, and then if we walk the objective of the microscope forward and backward, or if we move the sample forward and backward, and we take a series of pictures, we can slice through the entire structure, we can reconstruct that, and we can get a three-dimensional model. So this is great. This is actually wonderful. This is my career, but it still has those limitations. It still has the limitation of depth. It still can basically poison life if we use too much laser power, if we create too much light from these proteins within the tissue. So we still have these really big problems to overcome when we're trying to understand how these processes work and to try to be confident that we're not disrupting them in a way that we're really imaging or getting images of what we're doing rather than what is naturally happening. So many, many problems. This was a truly tremendous advance. Confocal microscopes were developed in the late 80s and really came into prominence in the 90s and the early 2000s. And they are still very much a standard of scientific research today. But again, they have their limitations. Well, now we get into the new stuff. Now we get into the really exciting stuff. Oh, yes. So some of these are, what we're looking at here are nuclei within a cluster of developing cells, some sort of organoid. This is what we had before confocal. This is what we have after confocal, looking one at a time, and this is a three-dimensional construction of all those nuclear bodies. And so basically what we were doing is going from here with basic fluorescence to here with three-dimensional understanding. Some of the things that I did in grad school were dissecting and imaging fruit fly developing eggs to understand how sheets of cells communicate with each other to form three-dimensional shapes like tubes within our organs and our bodies. So this is that process. And this is sort of the best that we can do with confocal when we're talking about live imaging, when we're talking about actually watching these cells. In this case, we have a fluorescent marker that is marking the outlines of these cells, and we're watching the construction and development of these tubes in a coordinated fashion. So this is an amazing thing to be able to do, but at this time, at least with the tools that I had, I was not really able to tell what was happening within those individual cells. Well, now we get to another realm of microscopy called the light sheet microscope. And before this, whenever we were imaging with a microscope, we had an objective, we had a series of lenses, and we were passing the light through those lenses, and we were reading the light that we were creating in these fluorescent molecules back through that same lens. That has certain limitations when you're doing that serial sectioning that I talked about. What happens is your resolution is really good end on, but when you turn the whole thing on its side, it's elongated, it's blurry, it's distorted. It's pretty close, but it's not really accurate. Well, when we started putting the light, the objective that was putting the light in perpendicular to the objective that we were imaging from and creating a sheet of laser light, suddenly we could get much, much better resolution in depth. Not only could we do that, but we could go much deeper into the tissue that we were analyzing. And through some properties of doing it this way, it was much less toxic. It was had much less potential for distorting the process that we were actually seeing. Rohan, how am I doing? It's cool. All right, we're moving well. So with these light sheet microscopes, suddenly we went from being able to view just groups of cells or maybe thin slices of tissue to being able to image whole developing organs. And in this case, we have embryos of different model systems. This is a chicken embryo in which the nervous system is lit up, and this is a flattened projection of a light sheet microscope 3D imaging the entire developing embryo. This is the embryo of a zebrafish, and we're watching here all the individual nuclei, all the individual centers, the data banks of the cells as they move around in the development of this organism. So we went from being able to image things on a cellular to tissue level to basically being able to image things on a tissue to organ to even organismal level. As another example, this is a developing fruit fly embryo. What is laid on your fruit that then starts to develop. And through light sheet microscopy, we were able to identify all the nuclei in these embryos and track them in real time and be able to watch the earliest developments and tissue movements of the fly that created the body plan. So now we have scaled up to being able to view things even on a, like I said, on an organ or even organismal level, to understand and see the interactions that really we had only gained parts of, but now we were able to see in totality. Well, they took that a step further. This is Nobel Prize winning Eric Betzig with me next to him in Singapore in 2017. This is me in 2016 sitting next to one of his microscopes. And what he learned how to do is create not just a sheet of laser light for light sheet microscopy, but a latticed sheet of laser light that could allow us to take the same strengths of the light sheet, the depth of imaging and the speed of imaging and the low phototoxicity of imaging, and now apply this, which we had used before on a larger scale, back down to a much more microscopic scale. And so with his lattice light sheet microscope, we are now able to track incredible events at incredible resolution. And another thing that he did, which is really interesting, being a part of an astronomy camp, astronomers have dealt with distant systems looking at very distant points of light for a long time. They've developed tools to be able to correct for the flickering and the movement that will distort our imaging of those. Something like that is, or one of those things is called adaptive optics. So astronomers will sometimes project a laser point onto the Earth's atmosphere or onto the moon to basically create a steadying point that they can correct off of to be able to sharpen up that image and stabilize it. Well, what Eric Betzig did and what other scientists did is take the imaging technology that we have, but employ that astronomy trick of adaptive optics. So when you're looking through all of this tissue and what you're trying to see is really distorted deep in, what we do is we project a guide star of laser light into that point. We then take an image of it, knowing what that light, because we're just projecting a point, we know what that is supposed to look like. We image what it actually looks like in its distorted form. We pre-distort the activation light going in, such that the image that comes out to us is perfectly corrected. To me, that basically sounds like magic. It goes pretty far beyond my understanding and my capability, but this is how we are basically taking tools used to image the stars and to image distant galaxies and turning them around. And so really all of this is just a matter of lens and perspective. A telescope and a microscope are fundamentally similar. We're doing many of the same things. We have many of the same limitations. So really, and I would also say that we have, what we think of as, almost infinite of variability and wonder out there in the cosmos, and I would say that we have just as much intricacy and variability inside, and that's really beautiful. So we're now getting much closer. We're being able to basically do this imaging without disturbing. This is work that my friends did at Stony Brook University in New York. This is vasculature or blood vessels of a zebrafish embryo, and this is a cancer cell that was injected inside, and this is the first time a cancer cell has ever been visualized, ever, in a living context, sticking, crawling, and then invading across the blood vessel wall into the organism. So we are able to capture these events now in near perfect resolution, in real time, without disturbing them, and that is really one of the most exciting advances in modern microscopy. Now, this is still on a relatively big scale. So scientists are also doing something called super resolution microscopy. In this case, we basically don't actually take a picture. What we do is we take all of these fluorescent molecules that we have put into the cell to mark different proteins and things, and we individually flicker them. And every time a single one of them flashes, we put a little pin at the center of it. So even though that flash is a big blur, we know that if we're only getting one flash at a time from one single fluorescent molecule, we can map where that molecule actually was. And so by doing this repeatedly throughout these cells and these tissues, now we're getting down much closer to single molecule resolution. Now we're getting down to something like 20 to 30 nanometers. This is incredible. 20 to 30 nanometers versus a micron, which is 1,000 nanometers, and a micron is 1,000th of a millimeter. Yeah. And now, finally, one of Eric Betzig's colleagues has taken this a step further. So we're now using the best that we have in the way of optics. We're using optical tricks to either move around the sample, move around the optics, put things in the way, cast new sort of micro shadows, and using computation, using algorithms to map down to the smallest level, the highest resolution that we've ever achieved, resolution that approaches electron microscopy, which can only be used on dead things, on fixed things, on embedded things. Now, in living contexts, we are able to get down to something like 5 to 10 nanometer resolution. And so what I'm going to leave you with are these pictures. This is a complex figure, but these are individual protein subunits of the nuclear pores that regulate what goes inside and outside of a nucleus in a single living cell. So we're looking at the surface of the nucleus, at the regulatory pores, the gatekeepers that are deciding what goes in and what comes out, and we're able to see those single subunits. We still have a little bit farther to go, but this is farther than we've ever been able to see, and with light microscopy, we can even see it in living contexts. So, thank you for coming on this journey with me. I hope I have conveyed how awesome and how complex the microscopic world is. And I hope that this encourages you to, at some points in your experience and in your lives, to regularly step back and look at the world and consider the world through a different lens. I think this is a very important part of our lives. I think perspective really comes from changing our perspective and reevaluating what we think. We're constantly doing that in the field of microscopy. We're constantly doing that in the field of astronomy, and we're constantly doing that right here in our lives. Thank you very much. It's been a pleasure to talk to you. I'll take any questions on the side.